Or I think we are live, right? Sorry for everyone that's staying on the YouTube channel. I think we have a little bit technical glitch. Just a moment. Apologize, and just to reintroduce the speaker. So first of all, welcome everyone back to the Early Cure Immunology Seminar Series. For those who are returning today, thank you for your continued support. My name is Zhu Zhou. I'm an assistant professor at Boston Children's Hospital and Harvard Medical School. Just to say a very few words about ECI before introducing Esther today. As you know, that ECI is a biweekly seminar aiming to increase the exposure for junior faculty working in the area of immunology. This year, we featured 20 speakers across the globe, and we have close to 30 recorded seminars on YouTube channels with hundreds of views. We are reaching over 800 email subscribers to Early Cure Immunology email list. Thanks all for the support and for the hard work from the community this year. The ECI will continue to grow next year to serve our community, and we will also need new blood to join our ECI committee. So please stay tuned for the email announcement and also Twitter account announcement. So now I'm really excited to introduce a speaker today, my dear friend Esther Florsheim. Esther did her PhD in the immunology at the University of São Paulo in Brazil, studying allergic inflammation in the airway. And also towards the end of Esther's PhD, she spent a year visiting the lab of Ruslan Mestov at Yale. That's also when I first met Esther back then. And she then returned to Ruslan's lab as a postdoc. Her work is actually one of the most fascinating stories about chasing the unknown in a scientific discovery. Not to spill her story today, but I can say that her work actually fundamentally changes how I think about what the immune system will do in our body. And Esther has received many recognitions, including the several conference travel award and the AI Young Investigator Award in the New England area. She has been invited for many national and international conferences already. Esther has been an assistant professor in Arizona State University since 2021. And I look forward to your seminar today and to learn about what your lab will conquer in the near future.
Hi, everyone. Let me know if you cannot hear or see my slides, but thank you very much for this incredible opportunity. I think this group is amazing. If there's anything good that came out of this pandemic, this is one of them. So let me start So the focus of this lecture today is to show you how mast cell activation upon allergen challenge drives brain responses and behavioral changes. I think one of the implication of this work is that allergic inflammation, at least at early stages, can induce protective mechanisms, which could represent the evolutionary reason why we have allergies. So the fact that the immune and the nervous system interact is not novel, and it has been studied for quite a while. But the mechanistic insights on, for example, precise nature of their interaction and their impact is still largely unknown. Or the types of neuroimmune modules, for example, there are so many different types of cells on both sides, right? Like different types of sensory neurons, different type of immune cells, how they interact. We know that they interact bidirectionally, but still the precise nature is missing. So one obvious example of this interaction between these two systems is the development of sickness behaviors promoted by the immune responses to infections. So these motivated behaviors include symptoms like anorexia, sleep, temporary depression, fatigue, and so forth. So anyone who got a cold or flu probably experienced these symptoms, and though you probably didn't particularly enjoy the experience, these responses were purposefully induced by the immune system to help you fighting the infection. So the immune system, the types of immune responses that we have, we basically categorize them in two different types of immune responses. What we call type 1 immunity is induced by things like bacteria, viruses, parasites. Those things are sensed through pathogen recognition receptors and induce what we call type 1 immunity. The aim of type 1 immunity is basically to eliminate these pathogens or to eliminate, to get rid of cells that are infected with these pathogens. We do have another type of immune response that we call type 2 immunity. This type of immunity is induced by things way more varied than the type 1 group, and this includes like toxins, venoms, macroparasite molecules, drugs, phytochemicals, allergens, and so forth. So how those things are recognized is still unknown, but the fact is that they all triggered what we call type 2 immunity. And the strategy of this type of immune response is to, instead of eliminate those things, because sometimes this is impossible, because it's a drug, for example, that binds covalently to your membrane, so you do not kill that thing. Instead, you try to detoxify or to remove this type of toxins. The other point I want to make in this slide is that all of those strategies aim to trigger a defense and to protect you from those external stimuli. However, if you trigger too much of a defense, you can also induce pathology. So for example, in the case of too much of a type 1 response, you can induce things like sepsis, perhaps autoimmune diseases. And in the case of type 2 immunity, if you induce too much of this type of defenses, that's what induces allergic disease and hypersensitivities. So we want to study this problem of allergy and type 2 immunity, and we decided to study this in the gastrointestinal tract, the main reason being that it's a major site in connection with the environment, and also it's a major neurological, immunological, and endocrine organ. So allergies can be induced by things like tree nuts, specifically food allergies can be induced by this major food allergens that we know of, like eggs, like peanuts, tree nuts, shellfish, milk, and so forth. Allergies in general, like asthma and other hypersensitivities, are induced upon mast cell degranulation after IgE sensing and cross-linking, as well as inflammation driven by Th2 cells and innate lymphoid cells type 2. And the symptoms of allergies can range from mild things like runny nose and rashes and watery eyes to things a little bit more dramatic like bronchoconstriction and intense diarrhea, for example, to even like life-threatening events such as anaphylaxis. So we know that allergies are exacerbated reactions of the immune system. Food allergies in specific, which is where we're going to focus our work on, is extremely prevalent, highly prevalent, and it's increasing. Nowadays, it's reported that 1 in 14 children in the US have some type of food allergy. The reason we have food allergies is unclear. It seems like the immune system should not induce this type of responses to something that is seemingly innocuous, like food antigens, for example. If you want to know a little bit more on what we think about this, you can access our latest review on why we think allergies are increasing and what those things are. Basically, we think that allergic defenses serve a purpose, and they are here because they serve this protective function, but they can be exacerbated in certain individuals that can induce then this detrimental effect of allergic, and that's the disease that we call allergies. One interesting thing about allergies is that a lot of the reactions and a lot of the symptoms that we know from allergies are actually basically like neuronal reflexes, basic neuronal reflexes that everyone can have, such as itch, vomit, sneezing, coughing, and etc. So there is this obvious relationship with the immune response and the neuronal activation. It's just that it gets exacerbated in allergic individuals. So going back to my first, one of my first slides that I mentioned about sickness behavior, it's pretty clear that things like viruses or bacteria can induce the type 1 immune response, and this affects your behavior, behavioral defenses, and you induce sickness behaviors, right? So this is what type 1 immunity is known to, how type 1 immunity is known to affect behavior. However, what happens in type 2 immune responses, like when you are dealing with something extremely different from a pathogen, for example? In this case, we don't know anything about the types of behaviors that are induced. And that's precisely our question. One of the works that motivated me to start this whole project was this that I'm going to show you here, this graph. This is from Denise Cara and Nelson Vaz, a Brazilian group, and they published this in '94. And the title of the article is exactly my title here, is the immunological induction of food aversion. And that was a very simple assay that they did. And basically they show, so this is on the y-axis, you can see milligrams per weight, basically, of the solution. This is basically how much mice are drinking from the solution. So, and the bars here show 3 different experimental groups. The first is like a normal unsensitized mouse or mice that eat or drink this amount of protein. Here's what they are giving is an egg white solution. So this is how much, if you offer a control unsensitized mouse, they like drinking from the solution containing this egg white antigen. However, if you pre-sensitize these animals to this egg white solution, this is what they are calling immune, then they decrease the amount that they drink from this bottle. And that is reversed if you previously tolerize these mice to this egg white through the oral route. So this shows something very simple, but very profound, in which the immune system can control the behavior, can affect the behavioral, in this case, of the preference of mice to this certain solution. They also showed a bunch of these other Brazilian groups that inspired me to do this work. I put all the reference here, but just another interesting example here. For example, on the top panel, I'm showing how mice that were sensitized to peanuts, when they were offered the option to eat from peanuts or from corn, they only ate the corn, but not the peanuts, and then vice versa. If the bottom panel shows mice that were sensitized to corn, but not peanuts, and they now prefer the food that they were not sensitized to, showing this sensitization induce an antigen-specific avoidance behavior. This seemingly happens to humans as well, in kids, in toddlers with potentially food allergies. Is very clear that they show avoidance behavior to the food that they are allergic to. And this is reported to be one of the main problems of giving oral immunotherapy, because more than 50% of the cases actually failed when you try to do oral immunotherapy, because you need to give tiny bit amounts of the allergen, and kids would refuse to eat or drink even that tiny little amount. So that happens in humans as well. So this is what motivated us to do this work. And the aims of this project was first to characterize this avoidance behavior in a food allergy model. The second one, to determine which central mechanisms are correlated with these behaviors. And the third, to identify the immunological mechanism and how this gut inflammation is associated with that behavior as well. This is how we induce allergic sensitization in mice, very similar. This is the classic model to induce that. Basically, we give on day 0 and day 7, we pair the aluminum hydroxide as an adjuvant with the ovalbumin as our antigen. We give this subcutaneously. And the behavioral response we're going to address by checking the preference test on day 14. So mice are here only sensitized, not challenged. They are only able to do that. This was done in mice, specifically this strain of bulb C mice, and this is the allergen that we used. This is more specifically how we did the behavior test, also called the 2-bottle preference test. So basically, after mice are immunologically sensitized, we individually cage mice in these cages that are called licometers, also because we have 2 bottles and each one of them have like a metal platform under them. So they can, every time they step on this metal platform and they lick from that bottle, this counts as, we can track the number of licks in each one of the bottles. So the preference test itself is giving either water, regular drinking water on one side, or the allergen solution which contains ovalbumin on the other side. And then we do like a water washout for like a day after the test. And then we switch the bottles to avoid place preference or place aversion. And we're going to find out how much they drink of each one of the bottles in the following slide. So this is the behavior shown by control unsensitized mice, or that were sensitized with PBS alone, with the aluminum hydroxide, which is the adjuvant alone without the antigen. This graph shows the cumulative number of licks over time. So how many licks over time they showed. In green, you can see this top part, how many licks, the cumulative licks on the ova bottle, so the bottle that contains the ovalbumin solution, and in blue, the one that contains the water, regular drinking water. So just by this graph, you can tell that control mice obviously have a preference to drink from the novel taste, which is the protein shake for them. However, if they were previously sensitized with ova now and the adjuvant, they switch their behavior a little bit, like they start drinking from the water bottle as the other ones, but quickly they recognize that this is something different, and then they start increasing their intake of water and decreasing their amount of ova intake. This can be quantified as area under the curve, and we call this percentage of ova preference. And you can clearly see that sensitized animals do decrease their preference to the ovalbumin solution. So after, on the second day of the bottle preference test, when we switch the bottles, we can see that the avoidance behavior to the ova bottle continues present. So this suggests that it's not a place aversion, but indeed a food aversion. Interestingly, this behavior happened very early on. Like if you analyze this data within just the first 10 minutes of providing the ova and the water bottle, you see that even within 10 minutes, you can already see a difference in the preference of sensitized animals, in which they are slightly less inclined to drink from the ova. This is surprising because as I mentioned, these animals were sensitized subcutaneously, so they have never seen this antigen through the oral route. So this is the first time that they see this allergen or this antigen, and they already show the slightly different preference within less than 10 minutes. If you try to trick them and you create a conflict by giving something that they really like together with something that they don't like, which is sucrose, for example, mice we know love sucrose. You can see it on the control animals that their solution intake increases. This is like total solution intake of the ova bottle, and the control increased by a lot because they really like sugar. But you see that the ova alum sensitized mice, even when you add sugar, you cannot reverse their avoidance behavior. This one is if you provide, instead of ovalbumin, if you give an albumin from a different species. So instead of egg albumin, if you give like a cow albumin or bovine serum albumin, that they don't have this avoidance behavior anymore, showing that this is antigen specific. And finally, we found that this phenotype is very robust over time. It's long lasting because even after 48 weeks after the last sensitization, even if you don't give any ova throughout all this time, they still maintain this memory of aversion somehow. So it's pretty long lasting. We did not test after that, but this is a very long time. It's half of the mouse's life. This is just a summary of all the characteristics, the general properties of this immunological avoidance, some of the things I already showed you, but just to recap. So this is dose dependent. If you start with a very low concentration of ova, they do not have avoidance. They start having avoidance after like 0.5, 1% of ova. However, if you do increasing concentrations of ova, if you start at 0.1, go to 0.5, and then 1, and then if you wait a week without doing anything with these mice, and then you provide the very low concentration of ova, they now become hypersensitive to even low amounts of albumin. That's what we're calling hypersensitivity. the memory and robustness I just showed you, as well as the specificity. The before last one, the adaptive characteristic means if we have now just one bottle and this bottle has ova, so they do not have a choice to drink from water or ova, they will still decrease the total amount, but that's their only source of water, so they will have to drink from the ova. If they do, they decrease, they have like weight loss if you do this for like time enough. That means that this avoidance behavior is likely adaptive. And finally, I'm calling this phenotype as general because if you sensitize mice with different adjuvants, for example, with LPS, that technically is not a type 2 adjuvant, but instead of type 1, type 17 type of adjuvant, you still promote avoidance behavior. So that shows that avoidance behavior is not a unique feature of type 2 immune responses. We also found if you collect the gut, the small intestine specifically, at that time that we see avoidance behavior, meaning on day 14 in only sensitized animals, we saw no evidence for, at least for cellular infiltrates or migration of any cellular, of any immune cell type. So you can pick your favorite there and look at that there is no difference in the presence of the cells, suggesting that this avoidance that mice have or mice expresses precedes any kind of gastrointestinal inflammation. So the fact that they have avoidance behavior is not a direct consequence of having inflammation, but probably the other way around. Finally, we looked at the brains of these animals when they were undergoing this experience of allergen exposure. To look into the brains, we did the same thing. We sensitized animals on day 0 and day 7, and then we gave some sham gavages with just water. And finally, we force-fed mice with ovalbumin, because obviously if we just gave them the option of drinking, they would not drink. So we wanted them to force feed with the amount that they would normally take. And then we analyzed neuronal activation in the brain, in specific areas in the brain. And we look at neuronal activation by staining with CFOS 90 minutes after collecting these brains. And these are the areas that we found to be differentially activated. The first one is the NTS, or nucleus of Tractus solitarius. The second one is the parabrachial nucleus, and it's a specific area within the parabrachial nucleus called the lateral external parabrachial nucleus. And finally, the central amygdala being extremely robustly induced by allergen exposure. So those areas are interesting when we saw them, because those are the ones that are known to be like classically induced by aversive stimuli, such as, for example, if mice drink like a bitter compound, or if you pinch the mouse tail, for example, those are the areas that will be light up. We obviously don't know much deeper the neuronal circuitry, if they are exactly the same type of neurons that are induced in allergy versus another aversive stimuli, we don't know. This is just a schematic with all the data from the CFOS activation within the areas that I showed you. So this is what we think is happening. There are these projections that are known to happen from the NTS to the PBN to the central amygdala. Central amygdala is a famous area that is associated with fear conditioning and anxiety-like behavior. So those are the pathways that we think are activated. We did not find other areas that had been implicated with this phenotype before, such as the parabrachial, such as the paraventricular nucleus in the hypothalamus or the lateral hypothalamus. How does this information get to the brain? Long story short, we don't know. What we can tell you for now is that after a very long experiments and trying to find out how this happens, our obvious suspect was the vagus nerve, since the NTS is one of the major areas activated by allergen exposure. We thought that the vagus nerve was relaying information from the periphery to the brainstem. However, we did subdiaphragmatic vagotomies, and we did not see any difference in the avoidance behavior or the preference to ovalbumin in the vagotomized mice. The vagotomy was confirmed by the presence of fluorogold in the dorsal motor of nucleus of the vagus nerve 2 weeks after injecting this fluorogold IP. So we do not think that there is a role of the vagus nerve so far. And we still don't know how this information gets to the brain yet, but I'll suggest one mechanism at the end of my talk. So going forward to the immunological mechanism, at this point, if mice are only sensitized to ova and alum, one of the only things that we know that is different immunologically speaking is that these mice have the induced humoral responses, meaning they make ova-specific IgE, as you see in this first graph here. Sensitized animals increase their production of, this is serum levels of IgE. They also have IgG1. So we decided to test, as had been previously suggested, that IgE can promote avoidance behavior. Using mice that are deficient in IgE antibodies, this is what we saw on the left graph is exactly what I showed you before, the allergic ova alum sensitized wild type animals. That's the behavior that they show, that they do not prefer to drink ova. However, if you do that in IgE deficient mice, they now like to drink ova as if they were control animals. And this is the quantification. So we think that IgE is one of the initial steps driving avoidance behavior. Interestingly, IgE has a very bad reputation for inducing the detrimental effects of the inflammatory response. So we decided to confirm what's the role of IgE. Now we did like a whole food allergy model. So mice would drive type 2 inflammation in the gastrointestinal tract to look at the role of IgE. One of those roles for IgE that is previously known, but we confirmed that IgE is very important for induction of diarrhea and transit time after oral exposure, as well as the accumulation of mast cells in the small intestine, and specifically in both lamina propria and the epithelial layer. So those are things that are dependent on IgE. However, we do have some readouts. Not everything is dependent on IgE in this protocol. So doing the same thing, for example, the systemic anaphylaxis that you can induce by giving systemic injections of ovalbumin, for example, intravenously injection of ovalbumin, that also has been shown that this triggers like a drop in temperature or hypothermia, which is a sign of anaphylaxis in mice. And it gets as low as you can see in this graph, as low as like 26, 27 degrees Celsius. This is the quantification on the left of the differential temperature shown by mice. And we see that IgE deficient mice have no difference whatsoever in this type of anaphylaxis. So the point is not everything is dependent on IgE for induction of inflammation. And as it is, this has been proposed as systemic anaphylaxis is likely dependent on this other antibody that mice have when they are sensitized to ova, which is IgG1, and IgE knockout have no difference in the amount of IgG1. So continuing, if we think it's IgE dependent, the idea is that IgEs sit on mast cells, so likely this phenotype is also mast cell dependent. So to test this hypothesis, we got mice from Pierre Launay and Gregory Gauthier at the INSERM in France. So these mice were called red mast cell and basophil because they are also reported, and you can conditionally deplete mice upon diphtheria toxin injection. So that's the model that we use. So by depleting mast cells after they have been sensitized to ovalbumin, we found that now this mast cell depleted mice reverse their preference to ova and do not show avoidance behavior towards this allergen anymore. And this bypasses the need for IgE. So this is downstream of IgE as they have the same serum levels of IgE in their blood. This is just a summary of the mechanisms that we have tried and think that they are not implicated in the allergic avoidance. So here we think it's not this avoidance behavior to ovalbumin is not dependent on histamine, on serotonin, on CGRP or substance P, prostanoids or PAR2. Those experiments were done in different ways. Some we pharmacologically targeted, and I put within the parentheses there how we address those phenotypes. But the point is that we don't think that there is any role for those mechanisms there in inducing avoidance. So what can be inducing this phenotype? Well, we found, interestingly, that this enzyme called 5-lipoxygenase or ALOX5 was expressed, especially in the proximal part of the small intestine, in the duodenum, differentially expressed and highly expressed in the alum sensitized mice compared to control animals. We found that this enzyme, which is actually a rate limiting enzyme for the production of leukotrienes, is dependent on IgE and mast cells. So it's downstream of that. So we're now thinking, okay, maybe cystainure or leukotrienes in general are responsible for this phenotype. We indeed found by doing a mass spec experiments, we found this is just one example of the leukotrienes that we found being induced by in the allergic gut. And this is completely dependent on this other enzyme called leukotriene synthase 4. And here's just a summary of all the pathways that we targeted. I'm going to show you on the next slide what we the data for those experiments. But basically, we can prevent, we can block the this enzyme 5-lipoxygenase using this drug called xylulton. We also used mice that are deficient in the leukotriene synthase 4, which is the major pathway to make cystainure leukotrienes, as well as either deficient mice for this receptor 2 or drugs that we can also use to inhibit or prevent the binding of these leukotrienes to this receptor. And here's the summary of the data that I will show you with all these pathways. The first one showing the xylulton, which is inhibiting 5-lipoxygenase. If you give this drug just a few hours before testing for their preference to ova, we saw that they now reverse their preference and they start liking ova, so they don't show avoidance behavior. Same thing for the other two. Those are deficient mice in either the cystainure leukotriene synthase 4 or the receptor 2. So that's how we think this pathway is happening, and it's definitely dependent on cystainure leukotrienes. Finally, we decided to look for a different molecule, and this molecule is called GDF15, or growth differentiation factor 15. This is a molecule that belongs to the TGF-beta superfamily, and the only receptor known for this molecule is expressed in the area postrema and nucleus of tractus solitarius neurons, and it's called GFRAL. And our idea to look for this molecule was that in our own lab, there was a paper by Harding Luang and Andrew Wang showing that this GDF15 is a major responsible for the physiological adaptation to a type 1 immune event, including blocking tissue damage and tissue tolerance, for example. And the other evidence is that this GDF15 has been associated with the induction of nausea, for example, by chemotherapy, and in species that can actually vomit, for example, in shrews, it was shown that the injection of this GDF15 induces emesis, anorexia, and weight loss. So that was a good candidate for us to investigate if this is involved in driving avoidance behavior to allergen. So we first found that GDF15 is expressed. Those are serum levels of GDF15 in wild type that are sensitized to ovalbumin. This production of GDF15 goes up in wild type, but is completely dependent on all IL-4 signaling pathways and IgE and the presence of mast cells. When we block this GDF15 prior to the ova preference test with a neutralizing antibody, we found that this neutralizing antibody now can prevent avoidance behavior. This is the quantification for that, showing that GDF15 might actually drive the avoidance to ova. You can recapitulate this phenotype by giving recombinant concentrations of GDF15. So you see a dose-dependent induction of aversion, and that again bypasses the need for mast cells, for example. And the most interesting thing we found with this GDF15, which had been shown to be coming from the kidney or liver, is that we actually did not, first we looked in the intestines, and first we didn't found, we thought that this could be coming from mast cells, but as you can see in this top panel, just looking in the duodenum, you have in green the huge presence of mast cells. The green cells are Fc-epsilon-1 positive, so you have accumulation of mast cells within the duodenum of wild type. As we mentioned before, the IgE, the accumulation of mast cells is IgE dependent, but then looking in the large intestine, we found no GDF15 in the, or at least coming from mast cells in the small intestine, but then we looked at the large intestine and we found very big amounts of GDF15 coming from the colon, from the large intestine. And the cells that are producing GDF15 do not seem to co-localize with the presence of Fc-epsilon-1, demonstrating that mast cells likely do not secrete this molecule. And this was done by this technique called RNA-scope, which is an in situ hybridization technique. And again, this GDF15 is also dependent on IgE. This is just the quantification to show you that the first graph here is the presence of Fc-epsilon-1 positive cells, and this correlates with the inflammatory response. Mainly, you have accumulation of mast cells in the duodenum, and you fall over across the gut. So that's a compartmentalization, and that correlates. That does not correlate with where GDF15 is coming from, which we found coming from specifically from large intestine. So we don't think that mast cells are making GDF15. So this is the summary of the story. The allergic sensitization we found induces very robust, stable, and long-lasting avoidance behavior. That this avoidance depends on mast cells, IgE, cysteine-leukotrienes, and GDF15. And finally, that allergen exposure induces those aversive central pathways, which is the nucleus of Tractus solitarius, parabrachial nucleus, and central amygdala. This is just the summary of this story that I told you. We obviously have to find out still a lot more on what's going on. For example, we don't know exactly how this signaling from the allergen sensing in the small intestine by mast cells and IgE and production of cysteine-leukotrienes, how this is related to this production in the large intestine of GDF15. It's possible that there is like some neuronal component on that driving this mechanistic response, but in any case, GDF15 we know can act in the nucleus of Tractus solitarius, so that's how we think this is being, this is how we're promoting this avoidance behavior. So just as a discussion point, I'm almost at the end. I think this is another example how the immune system can can interact with the neuronal system to drive appropriate behaviors that are important to deal with stimuli in the environment. So if you think about this, is nothing that different from like other types of sensory systems like olfaction, vision, and other classic sensorial systems that we have. So for example, all of them, you can either directly sense the stimuli. For example, in the olfaction, you sense the scent by detecting chemical signals directly by neurons, or in other sensory systems, you rely on specialized epithelial cells to translate this external stimuli into a neuronal response. And they all go to the brain, to the central nervous system to then trigger this behaviors that you need for whatever you're dealing with. So the last part, this last module, we think like the neuroimmune module, we think serves this exact same purpose. So in this case, for example, this neuroimmune module can sense noxious chemical stimuli, such as toxins, allergens, and other things. And that is reported to the central nervous system to also trigger, for example, what we showed is avoidance behavior. The system is more interesting in our opinion because it's very plastic. Not more interesting, but more, it's more plastic. It's not hardwired as the other systems are, but you can adapt to the environment on demand and make, as we know, the immune system is extremely good as inducing multiple and almost infinite types of specific antibodies, for example. Another point that I want to make before we finish is that I think this avoidance behavior that we saw is the type of defense mechanism that we were looking for that precedes the pathological outcome or the detrimental effects, which is actually the disease that we know. In this case, the disease being the allergic disease, but just to show that or suggest that, yes, the disease is obviously detrimental and you don't want it to get there. And this is unfortunate that happens to some individuals, but there is likely a protective beneficial component of this whole circuit. Like there's a reason perhaps that this pathway evolved or was selected for. So in this case, under these arrows, I can show you that, interestingly, the same mechanism that can drive the pathological effects on like a later phase, like for example, gut motility, accumulation of mast cells and anaphylaxis, the same mechanism, which is driven by IgE and mast cells, can also at an early stage induce protective mechanisms such as the avoidance behavior. And finally, that's, I think, one good example of the thing that we discussed in this perspective article 2 years ago, which we called food quality control. Just the point is that the immune system, as the other sensory systems, is also capable to select for food and trigger some type of behavioral, metabolic responses and so forth. So the immune system also functions as a controller of if this food is good or not for you, for example. And I think the take-home messages here for today's talk is that we found this neuroimmune pathway that we think is important for organismal adaptation in noxious environment. The second point is kind of what I just mentioned two slides ago about the protective role of mast cells and IgE. Again, those are two things. Like IgE and mast cells have really bad reputation for inducing anaphylaxis. So I think it's interesting that we're showing some more physiological response that is dependent on those two things. And the third one that I think is interesting to be considered from now on, how, again, in a different model, how we are seeing the immunological basis of behavioral responses. And I think that has potential impact for example, psychiatric diseases and psychiatric or mental illness. So I just want to thank you for the organizers, first of all, again, for inviting me, and obviously for everyone who was involved in this project. First, the Medzitov team, especially Professor Ruslan Medzitov. This whole work was done at Yale University. People from the Ruslan Medzitov, Nathaniel, Jamie, Schweling, and Fernando. We counted on an amazing team of collaborators who helped with all the behavioral assays and the brain responses. So the Marcelo Dietrich's group, including Bruna, Madie, Marcelo, and Carolina. Other professors like Andrew Wang, Hans Oetgen, and Pierre Launay, and Gregory Gautier, and finally Shushu Zhang and Stephen Lieberlis at Harvard. Thanks to our support from Food Allergy Science Initiative and the AJGMI. And with that, I want to thank you all for either being here or looking at this later, the recorded talk. I'm now at Arizona State University, and those are the topics that we're mostly interested in studying further, which are the physiological roles of IgE and mast cells, as well as this inflammation and behavior association. So thank you all. Contact me if you need or want.
Esther, thank you so much for this wonderful talk. For those of you who are on YouTube channels, please type your questions on the YouTube chat boxes, and we'll go through those questions one by one. And the first question is from a big fan of yours, Esther. So Josh Koning, the question is avoidance behavior in OVA LPS mediated by IgE? I think you mentioned that in the data already, but maybe you can just answer briefly so that we're all on the same page for this.
So yeah, very good question. It was the question if the OVA LPS induced avoidance is IgE dependent.
Yeah.
Yeah, it is. That's very interesting. I don't want to show the data, but it's on the article. It is dependent on IgE, which is extremely interesting because IgE was known to be just like a type 2 thing, but older, I think older reports already showed that some levels of LPS can induce some IgE, and we see the same thing. Like if you sensitize animals with OVA and LPS, you do induce a little bit of IgE. Is not nearly enough or not nearly as comparable to OVA and alum. So for example, OVA and alum, you get to like the microgram, like for example, 1 or 2 microgram per ml just after sensitization. With OVA and LPS, you get perhaps like 400, 300 nanogram per ml. So it's way less, but apparently this is enough to drive avoidance behavior, as IgE deficient mice do not show avoidance behavior when they are sensitized with OVA LPS. So that's very interesting that IgE is not uniquely part of a type 2 system, but it's probably like a much more general, and it suggests that maybe IgE, the main function of IgE might be to do some of these protective types of behaviors.
And the next question is from Philip, and he's thinking that has anyone looked at GDF15 in people undergoing oral immunotherapy or sublingual allergen administration? I think there's some observation that there's a high rate of dropout, maybe a correlation with the magnitude of the avoidance induction in these patients.
That would be really interesting. I don't think that has been done. That would be a super good idea to look at correlation of GDF15. And we know that this is important. For example, during pregnancy, the nausea induced in pregnancy is likely dependent on GDF15. And we know that GDF15 is usually induced by any kinds of inflammatory conditions, like cancer and infectious disease and autoimmune diseases and so forth. That would be a very interesting to correlate maybe even the amount of IgE and GDF15 that people have. Very good idea.
Yeah, really interesting. And stay on the GDF15 a little bit longer. So I have some questions, Esther. The expression pattern in the colon is really interesting. It's more towards the crypt other than the tip of the intestine. And do you know if other expression patterns or expression itself depends on microbiome in the colon?
Very good question, Shu. We still didn't do this experiment, and Nathaniel is actually trying to characterize by doing single cell which types of cells. It's seemingly, well, we don't have the full results yet, so we cannot speak much about that, but it's seemingly like a type of rare epithelial cell that looks like a goblet cell that is making this GDF15. And that would be interesting if that's microbiota dependent, which is very likely.
That's really, really cool. And another minor question, for the GDF15, have you tested whether the delivery route will make a difference in making those mice have behavioral response, say IP injection versus IV? And also, do you know if injection itself will trigger similar avoidance behaviors for these mice on the drinking water?
So the first part is if a different route of exposure. When you say a different route, is a different route in the sensitization or in the preference test?
A different route of delivered GDF15.
GDF15.
Right, either given by IP or IV injections.
We haven't tested that. We only gave IP, so presumably systemic is enough. That would be interesting to look at, like intranasally, for example, if that. We know, for example, that animals, and that had been published also by this Brazilian groups in like the early 2000s, that in a asthma model, like if you sensitize animals to ova and then you show if you like have ova in the airway, they also avoid they avoided the environment that has ova. So also the nasal, the airways can also induce avoidance behavior most likely. That would be interesting if we could give like intranasal GDF15 and also show that. I thought you had asked about the exposure. So let me just answer that also, because so if you give, we gave ovanalum subcutaneous, but if you give ova with cholera toxin intragastrically, which is a very typical way of inducing also food allergy or sensitizing to type 2 inflammation, you also show the same, they also have the same type of avoidance behavior.
And what about the other, in a way, that the other type of challenge that can make the mice have, in a way, bad memory, like say you stress the mice out, injections or bleeding, if those are conditioned together with ova, do you think that will also cause their avoidance behavior?
Yeah, good question. I think we have to do that, but that has been shown in the literature. Like if you pair, for example, ova, if you eat ova at the same time that something very stressful happens, like if you play like a really loud noise, or if you pinch, I forgot exactly the details how people have done that, but that was shown to have some association. So you're right, like that could be a typical Pavlovian conditioning. What we know from neuroscience field and this conditioned taste aversion is that everything that is through the oral route for some reason has a way more profound effect in pairing than any other place. So even if you don't pair like the allergen or the oral stimuli at the same time, like there are a few hours of window that you can actually pair those aversive, like the unconditioned stimulus with the conditioned stimulus, and even that works. That doesn't happen anywhere else. Like if you decouple those two things like timely, that's not going to work to induce aversive responses, for example. So only through the route, oral route.
Very nice. And there's another new question pop up about GDF15, just to bear with us a bit more. So I think Olivia Lou is wondering that what might be the mechanisms of GDF15 derived from colon to act on CNS?
Yeah, that can be easily explained by, it's a circulatory factor, right? It's in the circulation. And the areas that we saw that are activated in the brain can actually be outside of the blood-brain barrier. So they could potentially directly, like neurons in the aeropostrema and a few neurons on the NTS can detect things the same way as they detect, for example, gut hormones to induce satiety, for example. They can also be detecting like circulating levels of GDF15 directly and inducing this aversive areas. So that's how we think now, but I think we still need to find ways to better tackle these problems and find out, like we did not see a role for vagus nerve, for example, but maybe that's because of the tools that we used, or there's still potential for a role of the ENS, for example, the enteric nervous system, or even like dorsal root ganglia or trigeminal pathways, for example, that we have not addressed.
And Philip had a very, very interesting question regarding, similar to what you're thinking about, the neuronal mediated versus immune mediated avoidance. So he's curious that is there a role for the pain induced by rapid allergic response that feedback to create those avoidance, or does avoidance come from mere sensing the overproteins in the context of IgE?
That's a very good question. We want to answer that as soon as possible, because that's like if pain, like we showed that there is no correlation with the inflammation, but we don't know if there is like a pain response being triggered at that time that they have avoidance behavior. So it's a really hard, we were broadly restricted by the main strain of mice that mostly show the phenotype, which is BOB-C. So we don't have a lot of the fancy state-of-the-art neuroscience tools in this background. So we are now trying to do that experiment and see, for example, TRPV1 sensory neurons are important in this behavior. So far, we don't have any evidence that that's true, but that needs to have better experimental tools to address that. That's a very good point.
Another more questions about hypersensitivity from Philip. So do you think the food hypersensitivities that operate via very distinct immunological mechanisms to that of allergy, such as the celiac disease, will lead to also taste aversion?
Yeah, that's a good question. Like a different type of, I mean, the fact that our OVA LPS also induced a little bit of IgE that can also induce avoidance behavior, for us, it shows that this can be induced way more broadly and not just specific to type 2 inflammation, right? I would be very curious if in a different model, like the autoimmune is a very good idea, celiac and maybe others. What kind of, if in that case you do also have some small amounts of IgE, and if that can induce food aversion, I would guess that would be the case based on our LPS sensitization of mice, but we don't know. That would make sense at least to have something like that at place.
Yeah. And then the last question so far on the YouTube channel. So first of all, congratulations on amazing work. Have you looked at the impact of your model? For example, the food allergy may impact phenotype conditions, functions of the resident brain immune cells, such as microglia.
We did not look into that yet. The only thing that we try to find, and it's still inconclusive, but mostly if the blood-brain barrier is permeable when we have like allergic sensitization, for example, it does not seem the case. Like it doesn't seem that blood-brain barrier is permissive, because we were wondering maybe the IgE sensing of allergen instead of being peripherally is happening centrally. And mast cells were shown many times to be in the central nervous system, mostly in the meninges, though, in the dura portion, not as much in the parenchyma of the brain. We do not know about microglia. We still need to look for what's happening in the central nervous system in terms of inflammatory or allergen sensing per se. We don't think that the allergen sensing is happening there, but that could be an indirect effect of like the peripheral information being triggered there and having some role for those. So we still need to look at those things. Thank you for all your questions.
All right, I think that's all the questions on the YouTube today. Thank you all of them watching from YouTube with us. And then if you don't have time today, then you can check back on the recorded video by Esther. Thank you all. We'll see you in 2 weeks. Philip will give us a sign that when the recording's over. And then we have more of a backdoor chat. Still waiting for the sign from Philip.
That was fun.
I